And then if you guys have, I'm going to try and eliminate some of these other screens and make sure that I have the chat window open. So if you guys have questions, feel free to jump in, have conversations, and stop me, all right? So the beginning is just a review, right? Methylation is a methyl group. It's a carbon and 3 hydrogens. And it's just a reaction, right? It's adding or removing the methyl group to something. And that's methylation. And it happens over and over. It's like a million times, in a short period of time. This is happening all over the body all the time. And why do we care? Because it either starts or stops reactions. It turns on genes. It activates enzymes. One of the big things that it does in detoxification is in that phase 1 is taking those toxic elements and turning them from fat soluble to water soluble so we can excrete them. So, in that process, sometimes things become more toxic, and that's where we need to make sure that, we're getting through all the phases of detoxification and everything is available, and it's not just, not just tagging them for removal, but then not removing the trash. So obviously some of the more relevant methylation reactions that are going on is turning on those detox reactions, turning on serotonin and melatonin production. And then if you're over methylating, and we're going to talk a lot about over and under methylating today and what that means. Because we kind of started down that path in the first one, but I think we need to go a little bit deeper on this. And then, turning off reaction. So that might decrease things if you're an over-methylator, and then you're able to turn off the reactions, getting you to decrease some of the symptoms there. And methylation silences genetic expression, even in genes if you don't have a certain variant, right? So methylation is works as a control switch in the body for a lot of different genetic expressions. So you can have the genes, but you can have the variations in the genes, right? But are they actually on or off and turning and doing things in the body at a specific time, right? So just getting your genetics doesn't really give you the whole picture. So we kind of skipped over the side part last time, but this is literally just methylhomocysteine giving, back. Giving SAMe the methyl group, and then that methyl group goes out and donates methyls. So SAMe is giving methyls to make creatine and phosphatidylcholine and 200 other different reactions controlled by all these other genes. So then you come down here and you have homocysteine, which doesn't have a methyl. And then it comes back through here using the MTR gene, gets methylated again, and comes back up. So it's just a methyl, no methyl, methyl, no methyl, right? Do you guys understand what we're doing here? And
Kind of.
okay.
I think so.
So, I mean, really, everybody says methionine, right? But it's really methyl homocysteine. And this is adenyl methyl homocysteine. So, methionine, and you add magnesium and a little bit of energy, and you get SAMe.
Mm.
And then SAMe delivers the methyl group to wherever it needs to go, right? So you take the methyl group, SAMe delivers it to do all these different reactions in the body that happen over here, and you end up with homocysteine.
So
And
what is
go ahead.
SAH?
Where SAH?
Yes.
So that is that's an intermediary in here, and there's a whole nother pathway for that.
S-adenyl homocysteine.
Yes.
Without the methyl.
Right. Because it donates the methyl. So this doesn't have a methyl. And then this enzyme turns it into homocysteine, takes the adenyl off, and you just get homocysteine.
Okay.
Okay.
Then it moves from there without a methyl,
Right.
and then it's methylated again with,
And then MTR with
right.
folate or
MTR.
B12. So this is methyl, right? So this is B12 and this is folate. Gets back to methyl homocysteine, back to methionine. And it's just recycled
Right.
over and over and over.
Can I ask a question?
Mhm.
How come sometimes you'll find methionine, how do you say it?
Methionine.
Methionine in like amino acid supplements. Why would somebody want to take that?
It donates the methyl
It
group, right?
donates the methyl group.
Right. They're after the methyl group, right? Versus really the homocysteine.
Right.
So would taking that lower high hit homocysteine? I'm so confused.
Yeah, I'm not, and this is why sometimes I don't specifically supplement for genes, right? I'm more
Yeah.
looking at these green things. Like, do we have magnesium? Do we have zinc? Is mitochondrial function good? Are we getting good energy? Right? You need zinc over here also. So what happens, when we don't have this and we're not converting methyl homocysteine to SAMe? And, what's going on here? And if we don't have good function over here, that's how we can get a buildup of homocysteine. But this isn't the only way it's broken down. Homocysteine, we'll see later, there's a lot of different ways that we can break down homocysteine and a lot of different uses, right? Cysteine, right, goes to glutathione. Cysteine gets converted to taurine. Cysteine works in the sulfate pathway, right? So there's a lot of different things that cysteine is needed for. So, where is the disconnect? It's not just a methyl, right? So homocysteine is going to be able to accept a methyl group. That's where you get into the MTR, the MTHFR stuff, right? And then SAMe is really important over here.
How come there's no E on the end of the SAMe?
I have no idea. I always use it as SAMe. I don't know why it isn't on this one, but I always call it SAMe.
Hey, Jill, I have a question.
Mhm.
You know how when you scroll through Facebook or magazines or whatever, and you see, oh, look at this supplement. There's so many people out there that might see it and then just go to the health food store or wherever and say, I think I'm going to start some SAMe. What happens if just anybody does that and they really don't need it? Is it going to harm them?
It could. I mean, they could have reactions to it. Not everybody can tolerate it. If you're already over methylating,
Okay,
right, and
thanks.
you're taking a methyl donor, I mean, the thing is, this is so bare bones right here. There's so much when you open up the full pathway planner that I have on the mentorship page. This is just one teeny tiny part of this huge biochemistry.
Mm-hmm.
Right? This is one reaction that magnesium is involved in. How many magnesium reactions are there in the body? Right? Hundreds, hundreds and hundreds. So, there's 70 different ways the body uses methyl, right? Methyl genes specifically. So, tons of different things to consider and look at. But this is the basics, right? That this reaction, methyl homocysteine to SAMe to homocysteine back to, adding and subtracting from methyl homocysteine to homocysteine back to methyl homocysteine, this reaction is happening hundreds and hundreds and hundreds of times a second in your body over and over and over.
So most of it's not reversible. It looks like it only runs in one way, except for right there with your.
Right here is
That's
back
your
and forth,
donator.
right?
Yeah,
And then
that
there's then you can zip here using the BMHT
okay.
pathway right from homocysteine to methylhomocysteine without the MTHFR pathway at all,
Oh, okay.
using zinc as a cofactor with choline.
So even that, so if you've got somebody who has a really low B12, they're able to compensate by probably if they've got more choline in their diet.
Right. This is
Using
the
another
thing.
pathway.
There are people, right? There are backup pathways for almost every single reaction in the body,
Mm.
right? There are probably pathways we don't even know about yet. That's why, sometimes we, we look at all this stuff and we look at,
That's why we look at organic acids, right?
well,
Because
I mean, even looking at organic acids, and we have all the best ideas, and then we give somebody something and they react totally opposite to the way we think they should react. Because we don't know it all. There's no possible way to see all of the reactions and all of the genes and which ones are on and which ones are off and how the body's going to react in any specific situation. So, adding in one nutrient or one vitamin supplement or one whatever to cover all of the genetic variables isn't possible. We can guess, but then there's probably going to come a day when it stops working, right? Those are the clients that you've given them all the supplements and whatever, and then life happens and they're back because now they don't feel great on this supplement you gave them anymore. Because it's deeper. There's something else going on. So, but this is how methyl groups are transported. This is basically how it happens. So if there's not enough methyl groups to add to homocysteine, then homocysteine levels build up in the body, right? So we've said that elevated homocysteine is associated with all these different things. And so getting homocysteine down is something that we need to do if levels are too high. There was also a caveat on here that Jen mentioned in the second class that wasn't in the first, that high levels, when they're like 15 to 25, like when they're high, can affect hearing. So if you have clients that have different hearing problems, hearing loss, so high levels of homocysteine can affect hearing, but it is a reversible hearing loss damage. So that's something maybe to consider in some of your older clients to make sure that you're checking homocysteine levels. So where does homocysteine then get the methyl group so it can transform back into methionine? So the one pathway is from folate, right? Plus B12. You can always do it from TMG or betaine or DMG. Either of these pathways give the methyl group to homocysteine. Some people make better use of a pathway over another. So this is why a lot of times you'll get some of these supplement forms that have multiple precursors in together. Choline goes up that, right? We said that choline goes up that center pathway here, and you can use the choline pathway here.
Where is TMJ there? I mean TMG.
It's probably not on that first one.
Okay.
But the choline one is. And then MSM might be another methyl donor, but research is kind of not certain on that one. But it is definitely sulfur methane.
Muffle.
Something. Methyls. It's got two meths in it, so yeah. But there are a lot of other different methyl donors, we said, right? So this
Sorry, can I
is
ask something, Joe? It's Tammy.
yeah, what?
I was in a workshop with Carrie Jones, and she actually said creatine could also work as a helper in the in this whole cycle, and it would help with methylation. Have you heard of that?
Creatine. So on the first dot in here,
Yeah,
yeah.
in some. Yeah.
So Sam donates the methyl group and makes creatine, which
Yeah,
is this,
exactly.
yeah.
so maybe if you supplement
Yeah,
creatine,
exactly. It
then
takes
you
the
don't
pressure.
have to, yeah.
It takes the pressure off. So it's
That's
one
what she
of the
said.
methyl, what do they call it? And we talked about that in the first, where,
Conservation.
what was that, Deb?
Conservation. It
Yeah,
conserves.
it's a methyl conserver. Because the body doesn't need to use methyl to create it. Same thing with phosphatidylcholine, right? So if you take phosphatidylcholine or you take creatine, then the body doesn't have to use the methyl group to manufacture it. And that's where 80% of methyls are used from SAMe.
Okay.
Okay.
Thank you.
So if you consume these, you're conserving the group so they can go on and do the other.
Well, I just had a light bulb go off.
Yay! I love it. I actually had one for my client. We'll get to it, but all right. So where are we? Other donors. So on all these are ways to break down homocysteine, right? So homocysteine is going to other places. So homocysteine with B6 and magnesium is broken down these pathways, right, into uric acid. Cysteine is also used down here to make glutathione, and then the glutathione recycling happens here. Glutathione also is broken down into cysteine and glycine and then comes back up. So, I mean, tons of different pathways. And then we talked about the transsulfuration pathway over here, right? Where cysteine comes down this way with the sulfite and is broken down here. And this one needs molybdenum. So people with sulfur problems, people with low uric acid, because this is also molybdenum dependent, right? So when we're talking about things in this pathway, you can, if people are low or high in certain things, you can use those things as clues, right? If you gave someone molybdenum and they felt better, right? You can pretty much figure out that there was probably a blockage here, right? That they were low in this pathway. But maybe you have a uric acid number. So you can look at their uric acid, and if the uric acid is low, maybe it's down at 2.8, then you have a clue that that person probably needs molybdenum.
Is uric acid? It's not in this.
It's right here.
Oh, okay. And where's molybdenum? I
Right
don't
here.
think. Okay.
So this exo gene needs molybdenum to create uric acid. And for most people, right, that should just get metabolized out and be good. But for some people, it goes high.
Have we figured out the glucose factor? Can we see that in this pathway somehow and make that connection?
This is homocysteine and the transsulfuration pathway over here.
Yeah, but right, uric acid, if you have high uric acid, that like predisposes you to diabetic problems, right? So can we
Yes. There and in the pathway planner, I believe there's a whole glucose metabolism one. We can look at it after if you want. I can pull that pathway up, but it's not in the slides.
Okay.
Is the pathway planner something new you put in there
The
recently?
pathway planner is in the membership portal and it is under the methylation mastery tab.
Okay.
So you guys have the whole kit and caboodle, not just the slides here, but all of it. And it's complicated, right? But I mean, the thing is, we don't need to understand all this stuff, right? All these things here, you don't need to understand all of it, right? But what you're looking for is like these little clues for molybdenum and uric acid. I mean, that's a clue for you. If someone's low in B6, they're not going to convert homocysteine to cysteine. because they need it here and you need it here,
Right.
right? And you know, B6, it's all over the place, right? Here's zinc again. Here's manganese, right? Like, so over and over and over, we're all trying to figure out what all these genes are and are they on, are they off? But I mean, like, how many different pathways do we have where genes need these nutrients, right? They need these minerals, they need these vitamins, they need the cofactors. And if we're not getting them, things are going to get blocked. So everything is on a feedback loop. Everything needs cofactors. Everything needs energy, right? All these reactions, you know, need energy. So, you know, the argument forever has been that you just increase methylation based on genetics. And if you give them methylated folate, then methylation improves. But it's correct, right? On paper, biochemically, that's how it works. But you don't know, right? You don't know all the pieces, right? We don't have eyes on all 70 genes. We don't know which ones are, I mean, even if they have the variation, is it active? Is it not active? Is it getting, you know, shut off because of a different feedback loop? You know, we don't have enough of a picture ever to make a strong case for just blanket treating with methylated vitamins. So, you know, biochemically, we can improve methylation, but the problem is methylation affects many systems, and so does folic acid. So, or folate. They say folic acid in these all the time, but it's really truly folate. So, for instance, mood disorders, depression, autism, ADHD, OCD, schizophrenia often has undermethylation as a consequence, right? So, as what's going on, and then the consequence of that is that they have reduced activity of serotonin and dopamine. But giving folate, methylated folate, has a greater effect in lowering dopamine by a mechanism related to the enzymes. And we talked about that, right? The different SERT transporters and things that happen. So what you're doing is basically turning on the negative feedback loop. And what you're trying to increase, you end up decreasing because of the feedback loop. Because there's SERT reuptake transporter proteins and it messes everything up. So you really need to know for sure. And there's ways to do that, right? So we talked about using, increasing methylation improves serotonin production, but then they also accelerate the expression of those transport proteins. So they reduce serotonin presence at the synapse and lower the activity of serotonin. So they increase the production, but lower the activity of it at the same time. Because there's that beautiful feedback loop. And when we try and do something, right? How many times do we do that in the body? We try and do one thing, and we're actually, because of the body's protective balance measures, we do the opposite.
So when you say they lower the activity, do you mean they are lowering the sensitivity at the receptor site?
Mm-hmm.
Okay.
Yeah, I don't know the exact, I mean, there's an exact mechanism here, but that's the outcome. So there's more serotonin, but the expression of the SERT transport protein to get it across the synapse is lowered. Because the body's like, I don't want more serotonin. You're going to give me more serotonin, but you know what? We're going to put fewer seats on the little transporter bus. It's not going to get where you think it's going to go.
Well, that's why you actually get a lower serotonin level with SSRI.
So how, I mean, aside from symptoms in a client, how else could you know that this is going on?
So this is the two sides of depression. I think it's in the next slide. Jennifer. So
Okay.
when you treat depressions with folates, methylated or otherwise, it isn't advisable unless you know whether they're over-methylated or under-methylated. Because under-methylated depressives have high serotonin reuptake and a low level of serotonin activity, and over-methylated have low serotonin reuptake and high levels of serotonin. So, I mean, people are doing genetic testing for this, right? I mean, now there's like whole things where you can get your genes tested and find out what drug you need to be on.
Yeah.
So it's important that folates are not given without clarification of the methylated status. Nutritional therapy for undermethylated persons involves supplements that support the 1-carbon cycle. And I did put on our site, if you guys aren't familiar with the 1-carbon cycle, that's over there with the SAMe and how that all happens on the side of that first slide we did. There's a whole video that I posted, a YouTube video of somebody else like doing it with cartoons and lovely things. So you can get a little bit more in depth on the 1-carbon cycle.
So that's under methylation too?
So
Okay.
while we understand that genetic mapping helps us predict the likelihood that they're going to be important, it doesn't reflect the actual status, right? And we go back to the fact that we're trying to track 70 different genes and the status of them. So what we can do is use whole blood histamine and homocysteine plus a really detailed history to get to the answer. So low whole blood histamine is typically associated with high levels of serotonin. And then you can treat it with supplements that increase, not that we ever treat, right? But for educational purposes, that you can use supplements that increase SERT protein activity. And then high whole blood histamine correlates with low serotonin. Those people we treat with methylation therapy and restriction of folates, acetylators, and other nutrients. These must be done carefully since lowering serotonin of someone who is depressed with low serotonin will likely worsen their condition. Which is also why I don't generally try and treat by just giving them one thing or the other, right? And generally a good level of whole blood histamine that's balanced is going to be between 40 and 70.
Is that like if you just were to do a histamine, is that typically on serum versus whole blood?
You have to get it in whole blood.
So it's a specific order that says whole blood histamine versus
Correct.
just serum. Okay.
Not all labs do it too.
Right.
Okay. Good to know.
So what we did in the first level, methylation increases serotonin production. It reduces SERT transporter protein action. The 1-carbon cycle is that biochemical pathway that produces methylators SAMe and methionine. Folates are involved in the cycle, thereby increasing methionine and SAMe. Folates, whether they're methylated or not, have a more pronounced influence on increasing the activity of SERT transporters and thereby lowering the serotonin activity. Without proper testing, you should not treat depression with straight folic acid or folates. It's important to utilize a test such as whole blood histamine and homocysteine along with a good history before you attempt to do this.
It's always more
Yeah.
complicated than it looks.
It is more complicated than it looks, which is why I always go back to foundational things, right? Because this can get really tricky. It's not that you can't do it, but it's just so nuanced and you don't ever see the big picture. So it's more like throwing an arrow at a dartboard, seeing what sticks, right? Like, are we going to hit the mark or are we not? Like, I don't know.
You know what I find scary, Joe, is, and I don't know, I would think this would be possible, but what is that? Is it serotonin syndrome? Is that what it's called? That actually can be deadly? And I think, God, could we mess something like that up royally?
Well, I mean, there's always the, first do no harm, right? And we really try and do that. And I mean, a lot of this isn't because I want you guys to dig deep and get, really good at deciding when you need to and when you don't, but just to realize that we're never really seeing the big picture. We're not seeing all the moving parts. There's always something in a blind spot. So this was back in 2013 that people were saying this. Never look at or treat genetic SNPs In isolation, genetic testing must always be interpreted in conjunction with functional lab testing to evaluate whether a mutation is currently causing any problem. Just because someone has a SNP, right, just because it's present, does not mean it's expressing or even creating an issue in the body. So many people run out and get genetic testing and go, oh, I have MTHFR. Oh, I have COMT. Oh, I have this or that. You have no idea whether that's actually creating the problem that you have. Um, so SNPs that might be related to B12 and folate include MTHFR, right? So that reduces the ability to activate folate to 5-MTHF, which can cause a folate deficiency. MTR, which can create B12 deficiencies. MTRR, which is it's that one side one, so it's a slower regeneration of the B12. The FUT2, inability to hold on to it. TCN2 is the delivery of B12 to cells, could be limited. So these are people that you can bypass the SNP by using sublingual B12. COMT, methyl B12 may not be tolerated, so this is where you usually do the hydroxyl form. And then MUT, which is an extra adenosine B12, might be needed for people that have that, because that enzyme converts methyl B12 to adenosyl B12. So, here's the MTR and MTHF here. You really, trying to figure this out and where things go can sometimes be tricky. It really can. So,
Ben Lynch would have disagreed with you probably, but although he's come around a lot in his thinking, I have to say.
yeah. Well, I mean, and this is the thing. I mean, this all the SHE conference, all this pathway planner, this is all stuff he put together right there in 2013, 2014, 2015, right? It's not available anymore. Like, because we've learned a lot about epigenetics, about what turns these things on and off, about the cofactors and the feedback loops. And I've always said, the body is super amazing because there's always workarounds, right? The body figures it out. We go and do all these things, and you would think, once we start removing organs and all this stuff, that the creature would just die, right? Like, but we don't. Our bodies heal and figure it out and find ways around it. People with traumatic brain injuries, right, where they're paralyzed and lose speech, and their body, it's supposed to be a left brain activity or a right brain activity, but then the body creates new pathways and figures it out, right? So it is amazing. And I think what happens sometimes is we get a little, it's that I can't remember what's the bell curve where you learn a little bit and you think you know it all, and then you figure out you know nothing, and then you come back to some balance again.
Is that the Dunn effect?
Yes, the
Dunn
Dunner-Kruger.
Yeah.
Yeah. Effect, right? And I think we've kind of run that gamut with MTHFR and all this genetic stuff where we thought, once we figured out that we mapped the genome, we were going to have it all figured out. And then we realized that mapping the genome didn't really answer the questions. It just gave us more questions that we didn't know. And then we figured out this epigenetics thing, right? And genes are turned on and off. And even if you have a variation, I don't use the word mutation anymore. Humans with real actual mutations generally don't survive, right? I mean, there's those, Downs and some of the other ones, but usually there's some foundational piece that's missing that's making that variation happen, right? So there's ways, right? There's other things to look at. We need to learn more, and we need to look at everything from a different perspective. So let's talk about under and over methylation. And it's really all about neurotransmitter balance and how we're feeling emotionally. So when we have strong emotions all the time, what does that do to our body?
Put you in sympathetic mode.
Yeah. Is that a good thing?
No.
Short term, it's okay.
Short term, it's okay, right? But all the time. What happens when we're just strongly reacting emotionally all the time?
Your adrenals get overworked.
Yeah, parasympathetic.
So when we have them, the nervous system regulation is a major part of healing. But if a nutrient imbalance is affecting our emotions, balancing this may take precedence over unlocking trauma. So we have those clients, right, where they're so depleted that they have no chance, right, of getting into a healing environment, right? We have to, it's this warring halves, right? So there isn't anything wrong with being over or under methylated, but it may cause some sort of mental distress. Too much methyl and overmethylation results in a unique set of symptoms that are different than those resulting from too little methyl, people that are undermethylated. While undermethylation is generally considered less problematic than overmethylation, you may want to consider methylation and its potential to cause mental illness. So undermethylation, which is called histodelia, too little methyl. Essentially, they have low levels of SAMe, and SAMe, right, is our methyl donor. This can lead to perfectionism, high accomplishment, and high achievement. Now, why would this be problematic? Sounds good to me, right? 15 to 20% of these people experience problems as a result of undermethylation, and this can lead to low levels of serotonin and makes them very susceptible to depression. So overmethylation, which is called histopenia, too many methyls, it is very active in the brain, and too much leads to too much of a good thing. So this causes an overproduction of serotonin, norepinephrine, and dopamine in the brain. And in many cases, these can cause psychological problems, including reduced motivation, reduced libido, weight gain, and confusion. So methylation is that process where chemicals called methyl groups are added to various elements of DNA, proteins, and other molecules to help keep them in good working order. Histamine is one of the chemicals that requires methylation to be metabolized correctly. So this is why we're going to start getting into how does whole blood histamine and those levels fit into the picture here? Why can you use whole blood histamine? So if the histamine is undermethylated, it can begin to build up in the system. And when blood contains high levels of histamine, known as the histodelia by the Pfeiffer Treatment Center, the excess histamine is stored in the blood basophils. And in brain neurons. This in turn can result in low levels of neurotransmitters such as serotonin, dopamine, the chemicals responsible for making us feel good.
So are we also
Oops, sorry, go ahead.
So are we also saying that if people are just in a high histaminergic state, then that can result in low levels of neurotransmitters such as serotonin and dopamine? Whether the methylation status is adequate or not.
Well, it's a sign of methylation status. We can use the histamine to tell us what the methylation status in the body is.
Mm-hmm.
Well, I would think that if you have high histamine and you're using up a lot of your methyl groups because of methylation, then you're going to have symptoms in other organs of low methylation. Is that right? Because if you don't have the methylation groups available because they've been used up by histamine to do other functions, then you're going to have, would you have symptoms? I mean, if histamine's involved, would you have symptoms on the other side of maybe low methylation?
Well, you're going to end up with, what this is saying is if the histamine is undermethylated, right? If histamine's burning it up, right?
Okay, so, okay.
It's going to, eventually, you're right, it's not going to get methylated, and then the histamine's going to build up. So
Okay.
histamine is an important neurotransmitter. It's responsible for regulating pain, sensitivity, sleep, tear production, sex drive, and mood. So methyl and histamine compete with each other. When histamine is high, it's a sign of undermethylation. And when histamine is low, there's overmethylation. With too much methyl, the body will overproduce dopamine, norepinephrine, and serotonin, the activating neurotransmitters. And with too little methyl, the neurotransmitter levels are going to be low. And this is where I started thinking about my client. My crazy copper client. Copper is responsible for breaking down histamine. And when there's too much copper in our body, histamine can be excessively degraded, leading to low histamine levels. And at extreme levels, excessive degradation of histamine can cause paranoia and hallucinations.
Hallucinations.
Ta-da! Like, okay, so now I have to go back to the drawing board and think about my guy again and go, what is going on?
Can I ask a quick question about and maybe you don't know, but about where it talks about the tear productions with histamine? So therefore, if you had elevated histamine, are you going to have lower tear productions and dry eyes, or are you going to have it the other way around?
I would have to look it up.
Okay, because an antihistamine tends to dry you up.
Right.
So I'm wondering if an excessive amount of histamine would create an abundance of tears.
I mean, that's usually what happens.
Yeah.
When I'm having an allergic reaction. So that my eyes are watery and.
That's true.
Right. So, I mean, if you wanted to actually check the mechanism of that, but.
No, I just have a client and their Sjogren's is what they're looking at, although she doesn't have any of the blood work for that. So now I'm going back and thinking, well, maybe
Right.
could it be a histamine issue? But she has dry eyes as opposed to excessive tears. So I'm like, well, it doesn't fit that model. So
Copper.
what?
She's got too much copper on board.
Well, that could be
Too much copper, low histamine, right. Yeah, copper, zinc, ceruloplasmin. Time to get those. So this is what we're dealing with, right? So histamine, if it's balanced with SAMe, right? Is balanced. So you have a histamine connected to a SAMe. This is a really super balanced body, right? So if you're under methylating and you don't have enough SAMe, you're going to have a lot of histamine that's not being degraded. And over here, you have too much SAMe that's looking to methylate something. So you have to interpret the clues that you have about a person's biochemistry.
So what is the niacin down there? What is that?
Niacin's going to help mop up the extra methyl groups.
Okay.
In an overmethylator, you have to get the flushing kind. It's uncomfortable, but that's going to work. And then here, right, if you have an undermethylator, you could supplement with SAMe to bring them back into balance.
So this could be where supplementing with SAMe when you don't need it ends up being the problem on the right.
Right. Yeah, I mean, and that's the problem, right? Was it Lee who said it? People are just, or Kristen, people are just out there reading blogs, trying to tag, and they're trying whatever they can, and they're just taking supplements willy-nilly because they read somewhere that something was going to help them. So some of the methyl donors, vitamin C, P5P, zinc, methionine, SAMe, and even calcium and magnesium can be methyl donors. Oh, Lauren back in. You're here twice, Lauren. Um, so because of this, this is a quote from Chris Masterjohn. Because of this, I don't think that it's useful to say that you are an undermethylator or you're an overmethylator. I think what we want to do is we want to start from the place that says, let's take you as an individual and look at what might be off balance, whether it's an undermethylation or an overmethylation or both at the same time. And not how do we take you as this over methylator and lessen your methylation, or you as an under methylator and increase your methylation, but how do we take you as an individual, put things into your system that will help your body properly regulate and balance itself. And I think the practical approach, when you look at it that way, is different because you're not just trying to decrease something or increase something. You're trying to put all the pieces of the puzzle together that allows that system to operate smoothly. I think he just said it so well.
Now the problem is just figuring out how to do that.
Well, I mean, it goes a lot back to B6, magnesium, zinc, right? Where we're
All the cofactors.
all the cofactors. Is the mitochondria working? Are we, what do we have? Takeaways, but I mean, don't go crazy. I'm scrolling back, to this big, huge one that we had. Right? I mean, when you start looking at all the things, all the orange, which, that's going to increase activity of these things, all the purple, right? High cysteine, low SAM, high levels of GSH, low reoxygen species, right? All the things that are going, all the red. I mean, these are all the reactive oxygen species that are created when we're just doing things normally, and is there oxidative damage? Are we getting too many reactive oxygen species? Are we not clearing these things out? All the feedback loops, it's you really just need to start looking at the places where we can influence this. Having good mineral balance, having proper vitamins, right? Getting good digestion, making sure that we don't have toxins blocking things up.
I think
What chemical is MN?
Manganese,
Okay, yeah, that makes sense. Okay.
molybdenum, manganese, copper, zinc.
I think the uric acid one is a little tricky because it could be low it could be that it would be low, but if somebody's got a really high sugar carbohydrate diet, it's going to be high and you
Right.
won't be able to see that it's actually low.
Mm-hmm.
But that's just one piece, I guess.
And how much of that is surrounding all of these? What
True.
are I mean, this isn't the end all be all, right?
Yeah.
This is just trying to highlight homocysteine and where it goes in the transsulfuration pathway over here, right? I mean, but every single one of these is connected to a whole nother section of biochemistry.
Yeah.
So, you can't just look at one piece. It has to be, stepping back and looking at the bigger picture. Because when we focus too much on just one thing, we miss out. When we focus too much on MTHFR and forget that all of this is happening too. And MTHFR is just, this little piece over here,
Mm-hmm.
right?
That's the one
And
you hear that's the one you hear everything about.
right.
Yep.